Adil.
Thank you very much, Anji. Yes, sir, as Anji said, my name's Adil Khan. I'm a PI at King's College London. Right. So in order to survive, animals often need to enhance their ability to detect and then respond to particular behaviorally relevant stimuli. For example, on the time scale of hours to days, animals can gradually learn the behavioral relevance of novel stimuli. And in this example, for instance, this monkey has clearly learned the behavioral relevance of this bottle-shaped object and the reward it contains. In parallel, at a shorter time scale of seconds, as very nicely introduced in the previous talk, animals can also attend to stimuli that are behaviorally relevant at a given time. So for example, this boy who's clearly very able to attend to one object while filtering out a large number of other stimuli from his environment. Now, these two phenomena, learning and attention, therefore serve a very similar purpose, which is to enhance the animal's ability to detect and respond to behaviorally relevant stimuli. And both of these phenomena are believed to be dependent on cortical plasticity. And so the broad question that I'm interested in addressing is, how do both learning and attention change the information processing in the same patch of sensory cortex, right? So, and to address this question, what I will do is I'll first describe how neurons change their responses as mice learn to discriminate visual stimuli, and then how the same neurons subsequently change their responses as these animals switch between attending to and ignoring the same visual stimuli. Right, so to do this, so this is the behavioral paradigm which we developed, which is, so head-fixed mice run through this virtual environment. On the walls of this environment, we can present different stimuli. The mice are trained to perform a very simple go, no-go discrimination task. Whenever they see vertical stripes, they have to lick a reward spout to get a reward. Whenever they see angled stripes, they have to not lick. So this behavior is nice because it's very rapidly learned. Mice become experts in 7 to 8 days. And it's V1 dependent, which you confirmed by silencing V1 while the animals were doing the task. So that's great, but to really tell if V1 is really involved in learning the behavioral relevance of these stimuli, what we need to do is to record from V1 as the animal performs the task. And to do this, what we used is in vivo 2-photon calcium imaging. And so the experiment would look something like this. As the animal is performing the task, we record from this patch of cortex using 2-photon imaging, and at the same time recording behavioral variables such as the pupil diameters, the running speed, etc. So since we know that interneurons, and again, very nicely introduced by Doris earlier, we know that interneurons exert a very powerful influence on cortical processing, and that different classes of interneurons can play very different roles in cortical processing. So we wanted to identify these different classes of interneurons in our data set. And for this, we used a post-hoc immunostaining approach, in which at the end of the experiment, we would take extract the brain, slice it parallel to the imaging plane, and then immunostain the slice for PV, SOM, and VIP markers. And then using image registration, we can find the same neurons back in the originally recorded in vivo data during the behavior. So then at the end, we can go back to our data and say, well, this neuron was clearly a VIP neuron, this neuron was a PV neuron, this neuron was a SOM neuron, and so on. And so with this approach, we obtain this simultaneous activity from PV, SOM, VIP, and pyramidal neurons, and across this behavioral paradigm. So now with this data set, we asked, how do things change with learning? And the most relevant metric for us was the stimulus selectivity, which we defined as the difference in the neural response amplitude to stimulus 1 minus the response to stimulus 2 normalized by the standard deviation. So just a very simple metric. And we asked what happens with learning. And so here's an example neuron, an example pyramidal cell, which pre-learning is not very stimulus selective. It fires approximately the same degree to the 2 stimuli, but post-learning, it becomes very selective, firing mostly for one and not much for the other stimulus. So you can take this metric, average it across the population, and ask what happens across the population. And here what we see is over learning, SOM and VIP interneurons don't change significantly with learning, but we see a very robust increase in stimulus selectivity for pyramidal cells and for PV interneurons. So this was already a very interesting result for us. In particular, we were quite intrigued by the fact that PV interneurons increased their stimulus selectivity, which is kind of contrary to the dominant model of that PV cells are just broadly integrating their inputs from the surrounding pyramidal cells. We dug into this quite a lot. I won't go into this in detail. This is published. But what we think we find is that there's a reorganization of the recurrent connectivity such that there are pyramidal PV subnetworks which form, in which cells connect to each other more strongly if they prefer the same oriented stimuli. So I'll just skip over this, but ask, okay, having established these results, what happens with now attention, right? So to study attention, we use the following behavioral paradigm. We trained mice to go in blockwise between a visual block, which is exactly the same as the behavioral task I just described, that go, no go visual discrimination paradigm. And after 100 or so trials of the visual block, we switched to an olfactory block. And the task is a very similar odor discrimination, where it's odor 1, lick, odor 2, don't lick. But on a fraction of trials, we throw in the very same visual stimuli, which now the animal, it's completely irrelevant to the task and the animal has to ignore. And the animal, we don't tell the animal when the blocks transition, they figure it out through the changing reward contingencies. And these mice are extremely good at switching back and forth between ignoring and attending the same visual stimuli. So here is the behavioral data. The y-axis is behavioral accuracy. 2 is approximately 85% accuracy. So you can see in the olfactory block, they're performing very, very accurate odor discriminations while completely ignoring the visual stimuli. And just a few seconds later, when you switch to the visual block, the animals are very accurately discriminating between these same 2 visual stimuli. And you can make them switch back and forth as many times as you like. So mice are performing this behavioral switching very, very robustly. And then we can ask what happens to the neural selectivity during the ignore condition and the attend condition of this attention switching paradigm. So here's an example neuron. It's you can see that in the ignore condition, it's not very selective, and in the attend condition, it becomes selective. So clearly there's an ability of attention to induce selectivity. And you can again take this value of selectivity, average it across all neurons and compare ignore and attend conditions. And what we find is very similar to previously is this ignore that the somatostatin and VIP interneurons don't change their selectivity with attention. But again, we see a very robust increase in pyramidal and PV cells. And from just a few slides ago, you will notice this is very similar to what we saw with learning. An increase in pyramidal and PV, no change in SOM and VIP. So on average, the changes which we see during learning and attention are very similar across these 4 cell classes. And so the next question which we wanted to dig into is, of course, what is happening at the level of individual neurons? Do the same highly plastic cells drive both of these changes? Or in other words, if an individual neuron changes a lot with learning, will the same neuron change a lot with attention also? And since we had recorded from the very same neurons from pre-learning to post-learning to ignore to attend, we could directly address this question. And what we find very interestingly is there's absolutely no correlation between the amount cell changes its selectivity with learning and the amount that same cell changes its selectivity with attention. So selectivity changes are uncorrelated at a single cell level. And mind you, this does not mean that there are two separate populations, one doing learning and one doing attention. No, it just means that they're uncorrelated. So we confirmed that this is not due to any large scale changes in response properties, which means because from the end of the post-learning of the learning paradigm, it takes 1 to 2 days for them to become experts at the attention switching. So in that time, we compared that the post-learning, the selectivity alone in the post-learning condition versus the selectivity in the attend block of the attention switching task. And that was extremely correlated. So it's showing that the cells are still preferring basically the same stimuli. It's the delta of that selectivity which is uncorrelated. So and then we looked at, of course, at the interneurons, and they showed basically the same effect. No correlation of the amount, not much correlation of the amount that they change with learning versus attention. So essentially, the changes in average selectivity can be very, very similar on average across the population, but at the single cell level, these are occurring through independent populations. So this really leads us to believe that there's distinct mechanisms underlying learning and attention-driven changes, and what might these distinct mechanisms look like? We're looking at a number of places, but I have time to just go through one, which is how the response magnitude changes. So here what I've plotted is, if you take the response curve in the post-learning condition and subtract the response curve in the pre-learning condition, so post minus pre of the same neuron aligned to this one to stimulus 1. So this is what we call the difference PSTH. And this is each row is one neuron. And if you look at this first learning data, you can see that there's a large number of cells which are blue, which is negative. And negative values essentially means that the neuron reduces its responses to the same stimulus during learning. So there's a dominance of reduction in responses with learning. But with attention, you can see there's a bit of more of an even spread. There's some enhancements and some reduction. And so you can plot the histograms of just averaging from 0 to 1, and you can see this clearly large bump of negative values for learning, and attention more nicely spread across 0, in fact, a little bit more enhancement than suppression. Now, again, since we recorded from the same neurons from learning and attention, we can ask, if an individual neuron tends to be suppressed with learning, is it also tending to be suppressed with attention? And the answer is no, there is absolutely no correlation. Once again, the same neurons can do completely different things with learning and attention. So very versatile behavior depending on the behavioral demands. And so finally, just to zoom in on this really interesting subset of neurons, which we call recruited neurons. These are the neurons which start off non-selective and become selective, either over learning or with attention, right? So if we just take those neurons, what do we see? So here are those neurons which over learning, they start off non-selective by definition. They become selective. So we're just plotting the average PSTH of all of these, about 200 or so neurons. They become selective, not by boosting the response to one of the two stimuli, but rather by selectively suppressing the response to one of the two stimuli. But in contrast with attention, non-selective cells become selective by a combination of enhancement of responses and a bit of suppression. Similarly for the neurons, and this is for neurons which end up preferring stimulus 1, neurons which prefer stimulus 2, same story. Learning is happening entirely through suppression. Attention is happening mostly through enhancement and not much change for suppression. So essentially, learning is driven by selective suppression. Attention is largely driven by selective enhancement. And so this really underlines the fact that there are very distinct implementations of these attention-related increase and learning-related increase in selectivity. So just to summarize, what I've shown you is that learning and attention both increase pyramidal and PV selectivity on average, but distinct mechanisms underlie the selectivity learning across the changes across learning and attention. What we think is happening is learning is driven by selective suppression, attention is driven by enhancement plus suppression. So thanks a lot to the people with whom I did this work, our collaborators and funders, and I have a postdoc position available in the lab, so please write to me if you're interested. And thank you very much for your attention.
Thanks, Adil, for the great talk on learning and attention. A very thorough study about neural selectivity on for these two different tasks. We have time for one question. We have a question from the audience, Elizabeth. Have you ever overlapped the visual and olfactory cues?
Yes, actually we did do that, and the problem is it's a bit of a mess when you overlap them. The major problem, I think, is that the onsets of olfactory stimuli are never going to be as sharply defined as the onsets of visual stimuli. We did it nevertheless, and we never really looked into it too much in detail, but it's actually not so surprising that we don't really see too much of responses to orders when we're looking in V1. So it was not really that interesting ultimately. And so finally what we did is we just separated out the things completely. So we never really overlap them much in this analysis. We just kept this visual stimulus comes on, goes off, and then the odor comes on, just to keep it clean.
Okay, thanks. Maybe one quick question. Have you looked at changes in correlated variability between cells across learning and attention state?
Yeah,
more generally,
absolutely.
How does it relate to the study by Ni et al. 2018 that show a shared components in learning and attention?
Yeah, that's a great question. And this is exactly, so one of the things I mentioned we're looking at is in this direction. Here, so since we recorded simultaneously from PV, pyramidal, SOM, and VIP, we can actually look at correlation between every possible combination of cell pairs that way. And what we find is that here's the result essentially, that you have 10 possible combinations, and that's summarized in the square where the thickness of the line indicates how much change in correlation there was. And with learning, there's this dramatic widespread reduction in correlations across everything we measure, almost everything we measure, most strikingly between SOM PV, SOM pyramidal, and SOM VIP. So really SOM cells get decorrelated from the rest of the network. And in contrast with attention, we don't see much changes. You can see most of these lines are very thin, except SOM and VIP within their own population change. So SOM gets more correlated and VIP gets less correlated. And we think, and so I mean, we hope to put, once we have this preprint out, you can have a look, but we've got this model in collaboration with Claudia Klopat's group, which has come up with Kata there has come up with a model in which she's able to explain this difference, why we get this particular change on the basis of top-down modulations impinging on this very structured circuit. So yeah, I'll be very happy to share you to talk about this if I had more time.
Okay, thanks. That's great. We have just finished everything.